When I started in biology, I wanted to make a contribution to biodiversity knowledge and to biodiversity conservation. So for my PhD, I studied a small mammal species, the Findable, for which high genetic diversity had been identified. So I collected samples throughout its distribution area by collecting animals in the field and also by searching for information in public databases. But I struggled a bit with the information available, as not always it contained all the relevant data that I needed for my study. So that made me aware that it is very important when we share data to include all the information that we collect so that it is reusable for the future. I'm Joana Paupério, and I'm going to talk to you about FAIR data and Biodiversity Digital Twins. We are all aware of the biodiversity crisis and the need to monitor and protect biological diversity globally. In this map that was generated by GBIF, the Global Biodiversity Information Facility, we can see the density of available data on species distribution or occurrences. However, the areas of the globe that are identified as having the brightest density of data don't necessarily correspond to the areas of higher biological diversity. And this is because data is made available from areas where more efforts is being done in collecting data and where researchers are able to make that data available in the public resources. But this is only a part of the data that is globally collected related with biodiversity. The biodiversity domain includes several areas of research, from species detection to inventory, studies on their biological traits, molecular characterization, and also to the interaction with other species and with the environment. This results in several types of data that, although deriving from different methods, are all connected in a network. And what we can see now is what is called a biodiversity knowledge graph, and that has been represented by Roderick Page as a network of the entities that describe the core of the biodiversity domain. And this includes taxa and their names. It includes the information on the specimens that have been collected around the world, the localities from where they were collected, the images that have been taken, the sequences that have been produced, the results of data analysis as phylogenies, and also traits that have been analyzed, and last but not least, the people. And there are a number of resources that are available that hold this information on these different data types that go from catalogs of species as the Catalog of Life, journals for publications, Wikidata for people, image archives, sequence repositories as the BOL data system and INSTC, the International Nucleotide Sequence Database collaboration, museums that hold the specimens, and data aggregators as GBIF, and also more specific resources for phylogenies as the Open Tree of Life and the Plant Trait Database. Despite of all these available resources, is the biodiversity data fully available and linked in the network? Let's look at an example. Let's imagine that we are a team of researchers that are studying genetic diversity of beetles in Europe and trying to understand how this diversity varies across Europe and with the different environments so that we can model predictions for the future. Where would we get the data from? So we could go into taxonomic resources like the Catalog of Life and try to get a list of beetle species that occur in Europe. We could also go into a data aggregator like GBIF and collect a list of occurrences of beetles within Europe. And this would give us a list, as we can see in this image. Then we would go into the sequence repositories, as for example, BOLD and INSTC databases, and collect the sequence information that is available on those resources and that comes from the European region. In addition, we could go into more specific resources, as for example, the Open Tree of Life database, where we could see if there are available phylogenies in this group within Europe. Then we would put that information together, analyze it, and relate it with additional environmental information that we would collect from other specific databases according to what we would like to analyze. But would we be able to find all available data? Probably not, because not all data that is produced is made available and in a public way to the users. And for the data that is findable, are we able to access fully? Is there contextual information that is shared? For example, do we know where the specimen was collected? What are geographic coordinates? Do we know if there are other biological information available on that specimen? So can we interpret the data correctly based on the information available? Can a machine interpret this data, and can bioinformatic tools use it directly for analysis? Data may often be accessible, but sometimes it's hard to find, and sometimes it's difficult to interpret because it's not shared with enough metadata or in a standardized and structured way. And that is what the FAIR principles try to guide users towards. So what is FAIR? FAIR stands for Findable, Accessible, Interoperable, and Reusable. And what does this mean? So for data to be findable, it needs to have rich metadata that accurately describes the data and provides contextual information. Where was a sample collected? How it was collected? What analyses were made? What bioinformatics tools were used? How did we collect and produce that information? Also, this metadata needs to be shared in a way that it's able to be indexed and searched by a machine. Also very important is the use of globally unique persistent identifiers, as for example, an URI or a DOI that allows everyone to access it and maintain the data accessible towards the future. And for data to be accessible, what is it needed? So we need data to be made available in an open and free repository. If authentication is needed, it needs to be allowed, and the repository or database needs to use a standard communication protocol so that it can be interpreted by everyone and every machine. And also, metadata needs to be accessible at all times. Interoperability means that machine can interpret your data. So you need to use broadly applicable language that is interpreted by all. You need to use standard vocabularies. For example, if you are referring to a country, you need to use a country name that is within a list that is accepted by the overall community. And also, data and metadata should be linked to other information that is available. For example, if I produce a nucleotide sequence from a sample that came from a specimen, and that specimen is stored in a museum as a voucher, then the data, the sequence information, should have a link to that sample that refers to the identifier that it used in the museum, so that we can go from the sequence to the museum and eventually collect additional information that is available together with the voucher. Also, for reusability, as we mentioned, metadata and data needs to be very well described and associated with detailed provenance. It needs to meet community standards, so use community vocabulary, so it's easily interpretable and understood by all, and it needs to be open and hold an open usage license. From these principles, from the FAIR principles, there are a few terms that pop up and that we can see are very important. One of them is open resources. So data needs to be made, together with its metadata, accessible in open resources. Then it's the description around the data needs to be rich as possible, so that everyone has the information they need to interpret it. Provenance is very important. We need to be able to link the data and metadata to the people, to the methods, to what has been done in order to collect it. And this data needs to be shared following community standards so that we can understand it and interpret it even by machines. And then we need linking. We need persistent identifiers that can connect the information between the different data types. How does this relate, and why is this important for the biodiversity digital twins? The digital twin is a concept that is applied to several areas of knowledge and is defined as a virtual representation of real-world entities and processes synchronized at a specified frequency and fidelity. In very simple terms, a digital twin includes data from several sources as input that is used to develop a model or an analysis and then will translate into different outputs. In the case of biodiversity digital twins, the input data can be either biodiversity data that can come from several different data types, but also different types of data as environmental or even climate data. So what are the challenges that are associated with making this biodiversity digital twins fair? So if we're collecting information from different data sources, then these may have different data structures and different metadata requirements. And these may follow even different community standards. So there may be the need to map these standards and requirements so that they can all be communicated in a similar, understandable language. Then this data will flow into the model and into the analysis, and regarding the model, the pipelines and processes that are used need to be documented so they can be reproduced in the future. Also very important is to keep and preserve provenance information, documenting all steps of this analysis. In terms of outputs, these need to be made available publicly in public databases, and they need to be made available using harmonized and standardized formats so that they can be overall interpreted by the different communities. Also, this output data needs to be linked with the input data and with the descriptions and the documentation that describes the model and the processes that were used to arrive to that data. And all of this will make these outputs understandable by the overall community and reusable. So what are the solutions that are being used in BioDT? So BioDT is making efforts to map metadata requirements when different data structures are involved and to use standardized data and metadata. They are documenting models and processes as well as software in different relevant platforms. The workflows and datasets are being packaged in machine-readable ways using, for example, Workflow Hub or Arrow Crates. The data is being made available in open access platforms and repositories, and provenance is being recorded in every step of the processes. So if we think about the challenges and solutions in biodiversity DT, we understand that FAIR is not only for the data, but is actually for all digital objects, and that includes the data itself, but also the models, the software, the algorithms. So it's a much broad perspective that we should take on FAIR. This initiative that we have described here with Biodiversity Digital Twins, together with the work that is being done with the community in the development of standards and by the infrastructures in terms of data integration and in the development of tools to make it possible for researchers and users to share their data and to make data accessible, is contributing towards a fair biodiversity data space. However, there's still a lot to do in this sense. There's still a lot of integration needed and a long path forward towards this. And it's up to me, us, and you to contribute to it and make biodiversity data fair.